In Chapter 11, we are going to look at the fourth group of macromolecules. So, just to kind of review, there are four groups of macromolecules or biomolecules that we're interested in. There are lipids, which we've already covered, as well as carbohydrates, and then the last chapter we talked about proteins. And so, this is going to be our last chapter, and we're really going to focus on nucleic acids. Now, nucleic acids are things like deoxyribonucleic acid, which we know as DNA, and ribonucleic acid, which we know as RNA. And we want to put some definitions to a few things that you've probably heard before, but it's just always really good to review. And so the first thing we want to review is DNA. And DNA is the molecule in our cells that stores and directs information responsible for cell growth and reproduction. Now, DNA is found in the nucleus of a cell. And basically contains the genetic information in what is called a genome. So when we talk about genomes, it's helpful to talk about genes. And so one part of the genome is a gene, and that contains the information to make a particular protein for a cell. Now, the human genome is a collection of, say, all our genes that humans have. And so the human genome consists of approximately 20,000 genes. Okay. And different cell types are going to express different parts of the genome. Okay. And so nucleic acids like DNA and its counterpart RNA are big molecules that we refer to as nucleic acids. So nucleic acids are going to be things like DNA, which is deoxyribonucleic acid, and its counterpart RNA, right? And these are made up of repeating units called nucleotides. Now, these are really big molecules. They are polymers. And so they're biopolymers. They have these repeating units. And so over the next few slides, we're going to explore the structure of the nucleotide building blocks. And that's going to help us understand the structure of nucleic acids and genomics, and then the study of genes and their interactions. So we're going to jump into nucleotides. So just like proteins are strings of amino acids, nucleic acids are strings of molecules called nucleotides. Okay. Now, nucleic acids are created from a set of 4 nucleotides in a given sequence. So nucleic acids are created from a set of 4 nucleotides in a given sequence. All right. And nucleotides are going to have 3 components. So the three components are a nitrogenous base, now this is the shorthand for that base right there, but basically you're going to see it's going to have a ring structure, and it's called a nitrogenous base because it contains nitrogen. Then the second part is going to be a 5-carbon sugar. So this is a pentose, right? Because that pent prefix means 5. And then the third part is a phosphate group. And that's a phosphate functional group. So let's dive into some specifics here. There are 4 different nitrogenous bases that are found in nucleic acids. And what we see here is each of the bases. So there are 4 different nitrogenous bases. Ooh. And what we're going to find is each of the bases has one of 2 nitrogen containing aromatic rings. And these aromatic rings are either going to be a purine or a pyrimidine. You're going to notice here, and I want to point out, there's the nitrogens there. We call these nitrogenous bases, and the nitrogens here. The purine is a 5-sided ring fused to a 6-sided ring, whereas the pyrimidine is just the 6-sided ring here. Now, let's zoom in on purines for a second. DNA and RNA contain 2 purines. And the 2 purines are adenine and guanine. And you can see the 1-letter abbreviation code here as an A and then a G for guanine. Then we also have the pyrimidines. Now here, right, DNA contains 2 pyrimidines. Okay, it contains thymine and cytosine. RNA contains 2 pyrimidines, right? And those pyrimidines that it contains are uracil and cytosine. And so what we find here is that the uracil is substituted out for the thymine in RNA. And so you can see the structures here, as well as the 1-letter codes. Now, one of the things that's very helpful is that, for example, cytosine is in DNA and RNA, but thymine is in DNA only, and uracil is in RNA only. So, now, nucleotides also contain a 5-carbon pentose sugar. To distinguish the carbons in the nitrogenous bases from the carbons in the sugar rings, the prime symbol is added to the numbering of the sugar rings. So the prime, which looks like this, right, symbol is added to the numbering of the sugars. Okay, and so RNA contains the pentose ribose, which is the R in RNA. So we can see that here. Okay, so RNA has that sugar, ribose, and then DNA contains pentose deoxyribose, which is this sugar here. That's in DNA. Now, the big difference here, of course, is the deoxy, and I want you to notice right here at that 2' position, what we see is that there's no oxygen bonded to the carbon in the deoxyribose. So otherwise the structures themselves are identical, but that's what it's missing. Now, for these two components to come together, they go through condensation reactions. So a pentose and a nitrogenous base can join by a condensation reaction. Okay, and what happens here is that the nitrogen in the base, which is shown here, right, bonds to the C1 of the pentose, which is shown here. The water in blue from the OH and the H, the water is produced from the OH and the H shown here. And so now we have the pentose and nitrogenous base joined by a glycosidic bond, okay? And the product here is called a nucleoside. Okay, so that product is called a nucleoside. And this is formation of the nucleoside is the first step in the synthesis of a nucleotide. So what will happen then is once this is formed, there'll be a second condensation reaction. And so what we see here is we have a hydrogen phosphate group, and we just refer to it as phosphate for simplicity. And again, notice the OH and the H here in blue are highlighted to give us water through a condensation reaction. And what we form here now is a nucleotide, okay, through a condensation reaction. And the nucleotide is the building block of the nucleic acids, DNA and RNA. So, just kind of as a review here, a nucleotide contains a nitrogenous base, a sugar, and a phosphate. A nucleoside contains only the sugar and the nitrogenous base. So when we look at this and make some comparisons here, what we see with our nucleotides, which have the phosphate, the sugar, and the nitrogenous base, is in black here, it's adenosine monophosphate, and that's AMP, and that's what's in RNA. And then deoxyadenosine monophosphate is in R, and you're going to see in parentheses here where that H replaces the OH, and that's what we see in DNA. And so both of the names for what we see in RNA and what we see in DNA are shown with each of the bases that we encounter. So different people learn different ways. Some people like words, some people like images, some people like tables. The same information here is presented on a table where you have the base here in either RNA or DNA, the nucleoside name, and then the nucleotide name here. And so that's just, I think, helpful, depending on how you like to organize your information. Now, many nucleotides linked together form nucleic acids. And these are linked, those nucleotides are linked through phosphodiester bonds. Okay. And the oxygens, right, which we're looking at here, we're seeing, again, this kind of linkage. It's a condensation reaction. We're forming water. But what we're noticing here with this linkage, here's the phosphodiester bond that we're looking at. And you're going to notice that it's connected to the 3' carbon and the 5' carbon of adjacent sugar molecules. So here is one nucleotide, here is the other nucleotide, and they join to give 2 nucleotide units. We call it a dinucleotide. And again, that di prefix means 2. So there is our dinucleotide. Now, similar to what we saw with the primary structures of proteins, we also have primary structures of nucleic acids, and that's just the sequence. Now, these sequences can be designated using one letter abbreviations, right? And then the convention is that if a single nucleic acid strand is shown, it's being drawn horizontally, then the 5' end is on the left, right? So it's written the 5' end to the 3' end, where the 3' end is on the right. And so you can see this here. So free 5' end, and then there's the free 3' end. Now the way it looks is it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And that's that backbone, very similar to the peptide backbone that we saw. And then what sticks out are the nitrogenous bases here. So you see the C and the T and the G, the T, the C, and the A. And so then we could even shorten it here, right? But we shorten it even further to just show those abbreviations for the nitrogenous bases. But again, we go 5' to 3' end when we're writing. Now, a few historical notes here. Erwin Chargaff noted that the amount of adenine is always equal to the amount of thymine in a DNA sample, and then the amount of guanine is always equal to the amount of cytosine in a DNA sample. And so what we know from this is that the number of purines equal the number of pyrimidines in DNA. Okay, so he analyzed a lot of the nucleuses to figure this out. Now, this is primary structure information, but DNA also has a secondary structure a secondary structure, which is a double helix. And so DNA's secondary structure is described as a double helix, right? And this was first proposed in 1953 by James Watson, Francis Crick, James Watson and Francis Crick, Rosalind Franklin. They used Rosalind Franklin's data for this discovery. So she's credited, she took the pictures that showed us that this was a DNA double helix. So she is now also credited, but it took a while for people to realize her contributions, and she died before the Nobel Prize was awarded. So a DNA double helix can be kind of envisioned as a twisting ladder, where the two strands have bases in the center. So you're going to notice here that the bases point towards the center, right? Their backbones run in different directions. So the backbones run in different directions. And so what that means is if one strand goes from the 5' to the 3' end, right, which is what we see here, 5' to 3', the other strand goes in the 5' to 3' end. So the 5 and the 3's match up, okay? Each rung, so these are kind of like the rungs of a ladder here. Each rung contains one base from each of the strands, and the two of these bases are held together with hydrogen bonding. So you can see the hydrogen bonding here. Now, Notice that the pairs A and T are always combined, and then C and G are also always combined, and these are called complementary base pairs, right? And one of the things that you're going to see here is that adenine and thymine form 2 hydrogen bonds, while guanine and cytosine over here form 3 hydrogen bonds. So, right, one of the things to keep in mind here is that the DNA in one human cell can contain 3 billion, with a B, base pairs. So let's look at an example of a problem. So let's say that you were asked to write the base sequence and label the 5' and 3' ends of a complementary strand for a segment. And so the strand would be, say, it's written 5' A, C, G, A, T, C, T, 3', and you need to write the complementary strand. So when you write the complementary strand, you want to kind of match up the number, and then you think about what the complementary base pair is. So the complementary base pair for A is T, for C is G, for G is C, for A is T, for T is A, for C is G, for T is A, and then we go to the 5' there. And so that's one of the questions that you may encounter, is to write and pair that up so that you could see which 2 strands make up a DNA double helix. Now, because DNA has a helical twist, further twisting can cause result in what's called supercoiling, right? So we've got DNA, it's twisting, it's going to end up in some supercoiling here, which makes that DNA more compact. The DNA is separated into 46 pieces, supercoiled around proteins called histones, and you can see that there. And then all of these are packed into a chromosome, and that's the DNA tertiary structure. And so we have the chromosome here. So we've learned about the different components of DNA in this screencast. We've learned also about their primary, secondary, and tertiary structure. And then in the next screencast, we're going to learn about DNA replication, and we are also going to talk about mutations and viruses.